Okay, I'm very excited for the next speaker. He's an associate professor of computer science at the University of Saskatchewan. He holds his PhD from the University of British Columbia. He completed a postdoc at Stanford University with the NIH Center for Biomedical Computation. He leads the image and data analysis group for the plant phenotyping and imaging research at the University of Saskatchewan. He also directs the Biomedical Imaging and Graphics Lab focused on 3D modeling, image analysis, and deep learning for biological and biomedical applications. Last but not the least, being his student, I can say for sure, he's one of the coolest supervisors any student can have. Ladies and gentlemen, please put your hands together for Dr. Ian Stavness.
Thank you, Anupama, for the nice introduction. So today I'll be talking about some of the computer tools we're developing in PERC, specifically around analyzing outdoor images. And this really is a group effort from many researchers. So I'll talk about aerial imaging, some of the work we're doing on ground imaging, and then I'll end with some of the analysis work. And I'll talk about outdoor images in this talk in particular because in Saskatchewan, there's a lot of outdoors. So this is a video of Curtis Pozniak's wheat breeding program, and as you can see, it's a substantial size and quite a phenomenal effort to imagine being able to analyze all of those plots. But thankfully, we have good collaborators led by Steve Shirtliff and his team that have been doing a lot of imaging of these breeding plots, both wheat, the canola, and lentil breeding programs, and they're ramping up. So, over the 3 years, they're getting more and more data. This past year, they imaged 38 trials, over 300 flights, and now they're starting to fly this monstrous drone there on the bottom that has a 100-megapixel camera co-mounted with a multispectral camera, as well as hyperspectral imaging, so the size of the data is also increasing. And the goal that's set to the computer scientists that are part of PERC is to come up with per-plot images and analysis to give back to the agriculture researchers and the plant breeders. So we have a number of challenges with this. First of all, there's just lots of pictures. So at this scale, it becomes very hard to manage and very difficult to organize those pictures manually. It's very easy to sort of mix things up, lose data, and so we want to try to help with the management. It's important to get fast feedback if there's problems in capture, because there might be a particular time where you want to image, and if something went wrong with the drone or with the camera, you want to know about it. And then also for all of these pictures, generally we make orthomosaics, so a panoramic picture of the field, and this is a very time-consuming computational step. Here's an example of orthomosaic. So here, this little inset picture is one picture we call a raw picture from the drone. So you can see it's just a portion of the field. There's a few plots in there. Oh yeah, this is where it would be in the field. But in order to get this picture of the whole field, we need to stitch them all together, and this is what's called an orthomosaic, which you've heard talked about a couple times today. So in order to help with this, we've been working hard to develop some web-based tools as a pipeline to process these aerial images. So Carl Gutwin has been working very hard to manage a great group of research associates on the project. William, Christian, Stephen, Banani, as well as another person not pictured, Brittany Chan, who was with us for a while. And this is kind of an overview of what the current set of software would like. This is like the website you would come to, and you can see there's a few different tools built into this. So the first tool that you would come to that we give to Steve and his team is an upload tool. So this is really where they can dump all the pictures from the drone and help to organize them. So there's some sort of check boxes and things on the left so that we know, we can identify what trial it is. We sort of scrape information that we can from the pictures to help automate as much of that as possible. And then there's some initial checks that are done to make sure there's GPS tags in the metadata for the images, make sure that it looks right before it gets uploaded. There's been another team led by Ralph Dieters who's been really working on the data storage and access management of this. We're currently using the university's data store to store these images, and it's sort of a data lake model. So distributed storage, we can sort of place storage local to the compute or have more flexibility there, and then you can get pictures back or data back basically through URLs, through queries. And tied to the storage is also the provenance or access to the data. So they've also been working on blockchain technology to help control access and make sure that people have limited access to certain images. So this, we think, is going to become very important to move these or migrate these tools to, let's say, a commercial setting where the data security or the image security is very important. So this has also been a big effort that's been going on. Once you upload the pictures, that automatically starts an orthomosaic generation step. So we do use commercial software for that. We use Agisoft Photoscan, but the process happens automatically kind of in the background. So that'll crank away on one of our compute servers. We've also tested it on the Microsoft Azure cloud. And then after a certain amount of time, you come back and see the orthomosaic. So it requires very little manual input, which speeds things up. But just an orthomosaic is not all we need, because really we're interested in these plots, the rectangular regions of interest where you have a different treatment or different genotype. And so there's also a semi-automatic segmentation tool. So once you have your orthomosaic, if you can specify sort of the geometry of the trial, so how big the plots are and how they're spaced, you can click on the corners of the trial, and then from that we can overlay automatically where the plots are. So it's sort of manual, but you just have to click a few times. And then from the field book, we know what all the labels for all the plots are. So sort of quite quickly we can get these segmented plots. And then we can also reuse segmentation. So if you do this once, the next time you fly and you get another orthomosaic, it'll be geo-referenced, so we can overlay the previous segmentation of the next one. And if there's any problems, like you can see they're kind of misaligned, you can kind of graphically move over whole ranges, or you can edit particular individual plots if you want that flexibility. The other thing we're working on in our team is trying to propagate this plot information back to the raw images. So again, this inset is the raw image, and what we can do is, based on the orthomosaic, we can kind of see where all of those rectangular regions were on the original raw images. And this allows us to view the pictures, the crops of the plots in all of the images taken, because as the drone flies over, it's going to be taking multiple pictures of a particular plot, because there's lots of overlap between the pictures. And so if we have a particular plot, we can then go back and see, okay, here's all of the pictures that we ever took in this flight of that particular plot. So this could be good just to have more pictures of a plot, but also you can pick out good ones. So if it's partly cloudy, you can pick out a sunny day, or if it was really windy at 1 second when the drone was taking a picture, maybe you don't want to use those. So we have a grad student working on understanding how much differences there are in the raws and how much useful information there is in those images. So you heard Scott Noble talk a lot about the ground imaging. This is sort of the main thrust of imaging in PERC around very, very highly detailed ground imaging. In our group, we've been doing sort of more quick and dirty approaches. So last year I showed this, our GoPro. So this is sending people out to the field with a GoPro camera on a stick, walking over the plats. And we really did this just to get lots of pictures quickly, because we knew it takes time to get very well-engineered things. And I put up the photos. Ilya is the grad student that did this, and then the other, Theo, Karsten from Plant Science, and then Liam, who's pictured in the video. They've walked many, many miles through these fields with the GoPro, so I'm highlighting them for their efforts to do this. And these are the sort of pictures we can get back. So this is a stitched orthomosaic of a plot with the inset showing the level of resolution. This is for a wheat trial, a wheat field, canola, and for lentil plots. So again, as you saw from Allison's talk this morning, you get really good high-res pictures. And really the goal that we've set out with these cheap imaging is try to do this very inexpensively. So we use cheap cameras. You can use others, but basically capturing pictures on a camera, and quite fast to deploy. So you can kind of quickly go out and take pictures, make it easy to operate so anyone can do it. So we don't have to get, there's not too much training that has to happen, and make it easy to replicate. So if you have many trials, you can just basically buy a bunch of GoPro cameras and get pictures simultaneously. So this really quick and dirty approach to imaging comes with a number of challenges, as most of you that are in the imaging field will know. So we have very inaccurate GPS. We have just the GPS on the GoPro, which is really flaky. It's noisy at the best of times and totally non-existent most of the time. There's a lot of camera motion, much more camera motion than with a cart system, for example. If we have multiple cameras on the cart, they're not synchronized. Like, we don't do any there's no special electronics to help synchronize things. And there's no feedback at all. So, the guy's walking, and you can't see if things are working or not. So these are sort of the downsides of this cheap approach. And so we're kind of framing this as a trade-off in complexity at acquisition time or analysis time. So if you want a really low complexity acquisition, then you have a much more complex analysis. But luckily, we're in the business of image analysis, so that's why we're interested in these cheap to capture and more complex to analyze. Another challenge we found is particularly with generating orthomosaics from these very low-level pictures. So in a drone image, as we saw before, one picture from the drone will cover multiple plots, as you saw. But when you're really close to the ground with a GoPro, you'll be walking across, and there'll be multiple pictures of that one plot or range, for example. And you can see that pictured here. You can see all the little black lines are outlines of all of the pictures that have taken for this particular plot. And with this particular walking, they sort of walked up one side, taking pictures, turned around, and then walked back down the other side in order to capture that whole length of rows in that range. So particularly those sort of acquisitions are very hard to generate orthomosaics of, because you have problems of the top path was taken much earlier than that. So if there's lots of wind, there can be very big problems. And particularly later in the season, like this is a good example of an orthomosaic from late season, but generally those are much harder because the plants are tall, so there's more parallax, more changes in perspective between images. So this is a big challenge. So to try to address this, we've this year moved to a new imaging setup we call the ProTractor. It was very similar to the GoPro, but now we have GoPros on the back of a tractor, basically. So this is collaboration with Agriculture and Agri-Food Canada. And really, this is also a project that just came from necessity. So Sally Vale and Christina Enck and Scott Stacy, the technician there, built this for us sort of last minute so we could get pictures of emergence. And part of the thing that is that it's very sort of to tailor images to the particular setup of plots, which has been talked about as well. So this is really built out of necessity so that we can image some of the nursery trials at Ag Canada. And we actually have 6 GoPros on this, so there are multiple cameras so that we can capture a wider, we can capture that whole row at one time. And then we can drive, so it's much faster than walking and less tiresome. And then Josh, pictured there, also logged a ton of time on the tractor, which is pretty impressive considering he's a computer science student. So we're also training computer science students for future fields. And I have to put credit where credit is due, because this is inspired by Steve's PhenoQuad there with the 20-foot 2x4 hanging off the side of a gator. So this is the kind of general setup, kind of looking front view of the tractor. So we have 3 cameras pointing down. And then these are the example images coming off of the tractor. So you can see the middle camera covers most of the row here, and then we have 2 more just to cover the 2 ends. And this is we did this initially to try to help the orthomosaic generation so that we can kind of cover the whole row, and we thought this would improve things. But another way to avoid the orthomosaic generation step, which is still very computationally expensive, is just to try to pluck out rows as we go from the raw images. So here's a sequence of a particular camera, and you can see the row kind of moves up the image as the tractor moves down. And the challenge with plucking out rows is that you'll see the same row multiple times, because so that red boxed row shows up 5 times here. So we have to pay attention to that as well. And this is where the sort of more complex analysis comes in. So we don't have GPS data that's accurate, so we can't know exactly where the camera is. So instead, we have to do some analysis. Luckily, in early season, where there's rows versus soil plant versus soil is very distinguishable, we can do things like threshold or try to pick out the green. And this is computing the excess green on the image and summing along the rows. So you can see the chart on the right-hand side gives you sort of how much green there is along each row, and sort of does a nice job of picking out basically approximately where the rows are as we go through these pictures. Based on that, we can do a thresholding and then computing a center line. So here's the same picture now where we're thresholding the plants and finding their center line. And this helps us to correct the angle differences as well, so we can straighten the picture, for example. And then we can do a rough registration to get the overlap, so we can detect when we have sort of the same row appearing more than once. And so this is a similar process to you need for stitching or orthomosaic, but it can be very approximate or very rough and much faster. So here you can see there's blue rectangle coming in on the bottom. Those are the first time we see a row. And then the purple ones where we've sort of seen that row in the past, and then the red ones are the ones going off. So anyway, this is an approach where we can say, oh, we found a new row, and then we can identify which rows belong to the same, which row is the same in multiple pictures. So this is the result in the end now, so we can kind of pluck out the same row, and we know that the same row has come up more than once. It's a bit easier to see here if we just show the crops. So I'll just play this one. And you can see the multiple pictures of the same row coming, and then it's quite distinguishable when it flips between rows. In fact, you can kind of see the slight different change in perspective as the tractor goes over top of the rows. So again, this is a bit more complex way to get plots out for these nurseries, but it makes the acquisition very inexpensive. And then once we have this data, we can develop visualization tools, a website where you can look at these high-res pictures over time. So here you see the whole range on the top, and then zoomed-in versions that you can manipulate the zoom. This was with Carl Gutten's group. And then if you have, you can kind of drag that date slider there, and you see the progression of these same plants over the season. Great. And so I'll end with talking about some of the image analysis approaches that we're looking at the research side of our group. And a number of them involve deep learning approaches. And these really will be kind of an overview because most of the students have posters, so I encourage you to see the faces and then go find them downstairs and check out their posters. So we do use a lot of deep learning approaches. The mainstay of that is convolutional neural networks. And sort of the main reason for that is because we're focused on outdoor images. So if you have images taken in very controlled environments, a lot of traditional image analysis approaches work quite well. So thresholding greenness to segment plants, or picking particular features to pick out to analyze the images. With outdoor images, since there's much more variation, variation in lighting because of sun and cloud, variation due to wind, due to capture because you have a cart or a tractor bouncing around. This variation means that those hand-tuned image analysis pipelines can be very difficult. You have to kind of tweak that threshold for each time you go out to the field because there is enough variation. So approaches like convolutional neural networks, the main difference is that these techniques will try to learn the most appropriate features and sort of parameters like thresholds in order to accomplish the task that you've given it. And you do that by giving lots of training images to these networks in order for them to learn what are the best features and best parameters. So this is kind of an overview of what, let me go back here, of what a convolutional neural network does. But if you have a picture, basically the convolutional part means that we convolve filters, we pass them over the image. And there's multiple filters that do that. So you have kind of a set of features that you're learning. And then you do this many, many times at different scales in order to make some estimations. At the input, you have images coming in. You do many, many convolutional blocks as well as other downsampling blocks in order to make some prediction at the right side of the image. And this is kind of a general approach that can be used for many tasks. And then we've done this for a few different sets of image analysis tasks. We've done a lot of work in counting things. In particular, we've done work on counting leaves. And this was some of the early work mainly because there was leaf data sets that exist. So I definitely have to thank the International Plant Phenotyping Network for providing some public data sets that we could use to try to work on through image analysis techniques. So this is one of the things we found that was a challenge was the fact that there's very few plant image data sets out there. So Jordan Nummins, my PhD student, applied these convolutional neural network technique to make predictions of leaf count. So images come in, and then you get a scalar count out for leaf counts. And at the time, this was the best leaf counter there was. And so we have a Frontiers paper that describes, in general, these deep learning techniques, and then applied to some leaf-related phenotypes. And this was using kind of a larger data set from IPPN, and then we followed that up with another student worked on a competition data set. This was a smaller data set that was a bit more challenging, but a very similar approach, and he got second place in the competition. And then we've worked as well with the University of Calgary to help to improve these leaf counting through synthetic data. So one of the challenges we have in general for doing machine learning with plants is that we have small numbers of pictures. Most of the deep learning techniques use data sets with millions of pictures, whereas we generally have hundreds or thousands of pictures. And so we thought of using synthetic versions of plants in order to help give us more training data. So you can see here, there's real rosette images and then synthetic ones generated from software by the University of Calgary. And we did find that these improve the training of these networks, so there is value in them. We've also tried to take these counting approaches out to the field, so trying to count plants from outdoor images. And these are more challenging approaches because they're more variable, but also wheat is much more difficult to count plants in because of their thin and grassy. So this is images from the GoPro. You can see the zoomed in picture. And then what we're doing is from these small patches, we're trying to count how many wheat plants are in there. And so these are annotated by our collaborators in plant science. And you can see an example here where the annotator has shown us what they're counting. So they're looking for individual leaves and then sort of clumped up regions. They say, oh, there's probably more than one plant there because they're quite dense. But the label they give the image is just the number, how many plants are in there. And so we're just trying to learn that count here. But a similar workflow, in this case, first we sort of find those patches, but then we do that regression approach, the same CNN to count plants. And that worked reasonably well. We've also started working on counting wheat heads. Again, more challenging because the pictures are quite busy and the background is quite busy. And so we're trying to use approaches that better localize where the wheat heads are. So you can see these are pictures of wheat heads, and then the color map, heat map there shows the regions that the neural network is focusing on in the image to count those as wheat heads. So you can see that the red spots are the places that are being focused on, and those do seem to be regions where the wheat heads exist. So we're still working on this collaborative project with plant science folks. We have some students working on detecting plants. This is a slightly different problem now. We want to actually draw a little bounding box around the plants to actually find out where they are. These are pictures from the ProTractor. You can see this is the sort of data that comes in, so you have to actually annotate exactly where all the little blue boxes are. So this would be the data we use for training. And then we've tested a few different networks for doing this. These are the sort of preliminary results. So this is the test image we would stick in, and these are the results that we come back. So this one looks pretty good. It seems to catch a lot of the plants. The challenge here, even in these very, very early, these are Carinata plants, there still is a lot of overlap, even at the 4-leaf stage, so it becomes a challenging problem. And then we have other examples where it doesn't do quite as well. So it picks up some of the plants but misses others. So again, we're refining this work to try to improve the detection of plants and tailor the architectures for these particular problems that we have sort of smaller data sets for. We've done work in the area of classification, one project on classifying lodging from aerial images, so is lodging or not lodging, both in wheat breeding trials and also in canola. And this approach actually used hand-tuned features, but did very good. So we could get very accurate results for lodging detection. And we have a student working on early vigor, so trying to estimate the vigor notes that were done for the nursery Carinata and canola plots. So there's sort of a workflow here where we check different features. Again, this is a more traditional approach where we're saying features like distance from the center line or perimeter or area, and trying to do classification into basically 3 vigor ratings, 1, 2, 3. And we get reasonable accuracy, but there's a lot of label noise. Some plants that look to us to be vigorous, the same amount of vigorous have different ratings. So this is where we're working with the plant breeders to come back and try to see what they're looking at in these images when they're doing rating and what's the most important aspect of the phenotype to try to capture. Segmentation, so this is similar to what John was talking about of trying to separate soil from plants. And so we're doing this Some of the trials, in particular, Kirsten Bett's lentils have not a rectangular grid, so it's very hard. You can't just do that grid overlay. And so here we need to actually try to segment out all of the plots. So we have a graduate student working on that, and then in collaboration with a plant scientist student to try to analyze these lentil plots. And then we're also working on deep learning approaches for segmentation. So these use a similar architecture, CNN, but you have an encoder part and then a decoder part that sort of expands the image back up in order to get a probability of different classes. And so this we've started to look at doing this for automatic plot segmentation, so trying to improve or make the plot segmentation aspect fully automatic. And then finally, we have a student looking at generative models. This is a slightly different approach, a deep learning approach, where you're training a neural network to generate novel images. And in particular, sort of two directions we're looking at. So generating NIR images from RBG or RGB images. So the advantage here would be that if we have a model that can make good predictions about NIR from just regular RGB images, then you can fly a cheaper camera and still make predictions about NIR. And then also going from low resolution to high resolution pictures as well. So again, here the motivation is you could fly a lower resolution camera and still try to pick up more high resolution features from those. And again, these also require lots of data to be able to train them, so we're still exploring their effectiveness. Here you can see these are the two paired pictures here. One is generated, one is real. And particularly when we look at things like NDVI or thresholded NDVI, where we really need to say, okay, is this generated image good enough to make a breeding decision or an agronomic decision? We think that there's an advantage there. So to summarize, we're building these tools to help create a workflow for all of the aerial imaging that's happening. We're doing inexpensive ground-based imaging with more sophisticated post-processing in order to get per-plot images and per-plot analyses. And then we have a big team of graduate students working on different phenotypic analyses, many of which involve machine learning approaches. So we're very open to collaborating with other researchers or industry partners and happy to discuss these things. Thank you very much.